In the early 1900s, F.W. Went made the profound statement that translates as, "Without growth substances, there is no growth." Plant hormones have an important role throughout the plant kingdom. A considerable amount of documentation shows that extremely low concentrations of a plant hormone can regulate many aspects of plant growth and development, from seed germination through senescence and death of the plant. Auxins were the first class of plant hormones discovered more than 80 years ago. Since this time, 5 additional classes of plant hormones have been recognized: Gibberellins, Cytokinins, Abscisic Acid, ABA, Ethylene, and the newest, Brassinosteroids. Numerous advances have been made in the use of plant growth regulating substances on a practical scale, along with basic research at the biochemical, physiological, and molecular levels. Today, the use of plant growth regulating compounds in agriculture is on the rise and will continue to increase as more scientific information becomes available. I'm Dr. DeBusk, and this video provides a survey of plant growth regulating compounds and their many effects on plant growth and development. The main difference between a plant hormone and plant growth regulator can be summed by remembering all plant hormones are plant growth regulators, but not all plant growth regulators are plant hormones. This means that plant hormones are naturally occurring, and so are plant growth regulators, but that plant growth regulators can be both naturally occurring and synthetic, and therefore cannot fall into the plant hormone category. Auxins, the first class of plant hormones discovered, were initially found in human urine. Since this time, they have been isolated from numerous plants and are thought to be ubiquitous in the plant kingdom. The term auxin is derived from the Greek word auxin, which means to grow. Indole 3-acetic acid, IAA, is the only active auxin found in plants today, and is involved in numerous physiological processes in plants, including cellular elongation, phototropism, gravitropism, apical dominance, root initiation, ethylene production, fruit growth, sex expression, and weed control. Auxins promote a dramatic stimulation in cell elongation in detached plant parts. Typically, in these detached tissues, little or no IAA is found. Therefore, when these tissues are placed in contact with IAA, there are large increases in growth. The dramatic increase in growth promoted by IAA is commonly used in bioassays for quantifying auxins, such as the Avena curvature and Avena straight growth bioassays, where the coleoptile, or sheath protecting the young shoot tip, is removed, placed on agar, and the agar containing IAA is placed back on the shoot. Phototropism is the movement of a plant hormone in response to directional fluxes or gradients in light. The Chladni-Wendt theory provides an explanation of how the phototropism process occurs. This theory states that when higher concentrations of the plant hormone IAA occur on the shaded side of the coleoptile in response to light, there's accelerated growth in this region. Growth proceeds normally on the illuminated side, and this differential in growth causes bending toward the light. Gravitropism is the movement of a plant organ in response to gravity. Studying plant growth and development in response to gravity is important for many reasons, such as better understanding how a seed knows which way is up or down during the germination process, understanding how plants grow under weightlessness conditions in an orbiting spacecraft, and considering post-harvest problems with flower stalks that exhibit unsightly bending during shipment and storage. When a seedling is placed horizontally, it responds to Earth's gravitational force with an altered growth pattern. The stem bends upward against gravity, which is called negative gravitropism, whereas the roots bend downward with gravity, which is called positive gravitropism. The Chladni-Wendt theory states that an accumulation of the IAA plant hormone on the lower side of the stem and root results in differential growth. The accumulation of IAA on the lower side of the stem accelerates growth on this side, while growth on the upper side proceeds at a normal rate. This differential growth promotes upward bending. The roots are much more sensitive to IAA, so the growth rate on the bottom side of the root is reduced, while the top grows normally. This differential growth results in downward bending. According to the statoliths theory, when the plant is placed in the horizontal position, statoliths containing starch fall to the bottom of the cell, which causes changes in IAA gradients, resulting in differential growth. This differential growth enables the plant to perceive that it is in the wrong position and reorient its growth pattern. Long before regulation of growth by plant hormones was discovered, botanists recognized that the apical bud causes lateral bud suppression. A simple experiment to prove that IAA is involved in apical dominance is to remove the apical bud and replace it with gelatin plus or minus IAA. The plant receiving the gelatin block plus IAA should have its lateral bud suppressed, whereas the one receiving no IAA treatment should have lateral bud break. Auxins stimulate root initiation and inhibit root elongation. Auxins are the only class of plant hormones that majorly affect rooting, and they are used commercially to stimulate adventitious rooting. The two most commonly used auxins for the promotion of adventitious root formation are indole 3-butyric acid, IBA, and naphthalene acetic acid, NAA. Many factors have been shown to induce ethylene production. In 1935, P.W. Zimmerman and Frank Wilcoxon were the first to show that auxin stimulated ethylene production in tomato plants. They treated one tomato plant with auxin, treated the control plant with water, and then placed both plants into a bell jar. Within a brief period of time, both plants became epinastic, downward bending of the petioles and stems, showing that auxin was not responsible for the change in plant growth and that ethylene was most likely responsible. Today, many of the responses once attributed to auxins are now found to be the result of ethylene. For example, auxins were sprayed on pineapples to stimulate flowering. Now it is known that the ethylene produced in response to auxins is responsible. Auxins promote increases in fruit size, which was shown dramatically by J.P. Nitsch's work in 1950 with the strawberry. In this study, no fruit growth was observed when all the seeds, auxins, from the strawberry fruit were removed and lanolin paste was applied to prevent desiccation. When seeds were removed and lanolin paste containing auxin was applied, fruit growth occurred normally. To show the involvement of seeds, Nitsch removed all the seeds from the strawberry fruit except for one, and the growth of the fruit occurred below the seed. Therefore, a correlation exists between seed development and the final size and shape of the fruit and exogenous applications of auxins to the fruit-induced growth. Auxins alter sex expression in plants. When it is sprayed on cucurbits, okra, pepper, and cannabis, there is a dramatic increase in the number of female flowers. Auxins also play a part in weed control. The first selective herbicide was not found until the early 1900s, and today more than 130 selective herbicides are used throughout the world. In the United States alone, 1 billion pounds of pesticides are sold annually, and over 65% are herbicides. Selectivity is the key to the widespread use of herbicides. Commonly used selective herbicide is 2,4-dichlorophenoxyacetic acid, 2,4-D, which is an auxin that is used to control broadleaf plants and lawns. Most, if not all, weed and feed fertilizers used by homeowners contain 2,4-D as the herbicide. Gibberellins are a group of plant hormones responsible for growth and development. Japanese farmers observed that their rice plants were taller earlier in the growing season. However, as the season progressed, these plants became spindly, chlorotic, and devoid of fruit. Japanese farmers called these symptoms the bakane, foolish seedling disease. It was later found the active component secreted by fungus was a gibberellin. Gibberellins are involved in numerous physiological processes in plants: stem growth elongation, bolting and flowering, seed germination, dormancy, sex expression, fruit growth, and parthenocarpy. More than 100 gibberellins are known today. Gibberellic acid, also called GA3, is the most commonly used. There are no synthetic forms of gibberellins. Many dwarf plants that are used in landscaping are short because the plant either cannot produce gibberellins or its cells are not receptive to the hormone. Dwarf trees like the bonsai seen here have highly reduced amounts of gibberellin. The stem starts to grow when it is applied in low concentration to a bush. The internodes grow so long that the plants become indistinguishable from standard plants. The gibberellins overcome the genetic limitations in different dwarf varieties. Gibberellins have been shown to stimulate bolting, which is rapid stem elongation and and flower initiation, which refers to internal physiological changes in the apical meristem that lead to the visible initiation of floral parts. Early researchers working on flowering showed that following the perception of light by the leaf produced a signal that, when transmitted to the bud, caused flower initiation to occur. A considerable amount of evidence supports that gibberellins are directly involved in controlling and promoting seed germination. Some seeds that are sensitive to light, such as tobacco and lettuce, exhibit poor germination in the absence of light. Germination begins rapidly if the seeds are exposed to the sunlight. If the seeds are treated with gibberellic acid, the light requirement can be overcome. Gibberellins cause the conversion of starch to sugar in the endosperm. These sugars are used for energy during the germination process. Also, the buds that are formed in autumn in trees stay dormant until next spring. This dormancy can be overcome by treating them with gibberellin. While auxins stimulated production of female flowers, gibberellins stimulate the production of male flowers in some plants. Gibberellins have been shown to affect fruit shape. When gibberellins are applied to a Thompson seedless grape bunch, it becomes elongated and the berries become larger. Gibberellins have been shown to promote the elongation of apple fruits to make the fruit look more appealing. Parthenocarpy is the development of fruits without pollination or fertilization, resulting in seedlessness. Gibberellins are commonly used to induce parthenocarpy in grapes. Cytokinins, like auxins and gibberellins, were also not initially isolated from a plant source. In 1955, the identification and purification of kinetin from aged herring sperm DNA was reported, and in 1963, the first naturally occurring cytokinin was found in plants. Cytokinins are involved in numerous physiological processes in plants, including cell division, organ formation, senescence, stomatal opening, lateral bud break, and sex expression. The major function of cytokinins in plants is to promote cell division by stimulating the production of proteins needed for mitosis. Cytokinins promote callus formation in most plant tissues. Callus is an undifferentiated mass of cells, and it is grown for tissue culture. The capability to regenerate plants from callus is a biotechnological tool commonly used today for selecting plants that are resistant to drought, salt stress, pathogens, herbicides, and others. For example, if herbicide-resistant plants are desired, callus is grown on toxic levels of a specific herbicide. Large numbers of Petri plates containing callus are grown, and most will die. However, when a small patch of callus proliferates on a plate containing toxic levels of a specific herbicide, it is selected. This callus cell line is grown and then transferred to a plate containing an auxin to cytokinin mixture to induce the formation of plants with roots and shoots. These plants are then grown to determine if they are resistant to the herbicide. Cytokinins can delay the senescence process in plants. When cytokinins are sprayed on a single leaf, the plant diverts nutrients to that particular leaf. This is called creating a sink, which delays senescence. Foliar applications of cytokinins have been reported to stimulate photosynthetic rates in a variety of plants. Some suggest that when cytokinins are sprayed on plant leaves, the stomates will open wider and stay open longer, which leads to an increase in photosynthetic rates. When cytokinins are applied to the whole plant, they will overcome apical dominance, thereby promoting lateral bud break. When plants are treated with cytokinins, the number of female flowers increases per plant in cannabis. A commonly used synthetic cytokinin is kinetin, and a naturally occurring form is zeatin. Abscisic acid, ABA, has been shown to be widely distributed within the plant kingdom, and it is the only inhibitor in this category. The major effect of abscisic acid in plants is its ability to signal that a plant is undergoing water stress. When a plant undergoes water stress, ABA levels are increased dramatically. The increase in ABA levels leads to stomatal closure, thereby preventing water loss via transpiration. In addition to being a plant stress signal, abscisic acid is also involved in dormancy, abscission, delaying seed germination, and retardation of plant growth and development. The ancient Egyptians gashed their figs to stimulate ripening, and the Chinese burned incense in closed rooms to enhance ripening. They unknowingly were treating their fruits with ethylene to promote ripening. In 1935, William Croker and his co-workers were the first to report that ethylene was the fruit ripening hormone. Other roles of ethylene in physiological processes in plants are its effects on seedling growth, senescence, abscission, and sex expression. Ethylene is the only gaseous plant hormone. When ethylene is applied to fruits, it causes them to ripen. For example, green tomatoes are shipped from California to Pennsylvania supermarkets where they are treated with ethylene to cause them to turn red. However, problems with flavor arise when this is done. In 1901, a Russian scientist was the first to show that ethylene has an effect on seedling growth. This scientist demonstrated the triple response, showing that ethylene inhibited elongation and promoted lateral expansion and horizontal growth. Ethylene can inhibit or promote elongation of stems, roots, or other organs. The inhibition of elongation is rapid and reversible. The triple response promoted by ethylene can act as a survival mechanism in seedlings. For example, when a seedling encounters a barrier such as soil crusting, a rock, or something that restricts its emergence, the seedling responds with the triple response. This allows it to either break through or go around the obstruction, thereby permitting the seedling to reach the soil surface and grow. Senescence is the general failure of many synthetic reactions that precedes cell death and is the phase of plant growth that extends from full maturity to death. Senescence is characterized by chlorophyll, protein, and nucleic acid degradation, as well as many other factors. Ethylene promotes senescence, yellowing. As plants age, ethylene is produced, which causes plants to senesce normally. However, when plants are stressed, ethylene is produced, resulting in premature senescence. Abscission is the separation of an organ or plant part from the parent plant. Ethylene promotes abscission in plants. The process of abscission is very important in agriculture because abscission or non-abscission of flowers, fruits, and leaves influences yields, efficiency, and harvesting operations. Ethylene has a natural role in the regulation of the rate of abscission. Abscission can be beneficial by facilitating mechanical harvesting, or it can be detrimental by resulting in premature abscission of leaves and flowers. Also, ethylene promotes female flowers in cucurbits and cannabis plants. The newest class of plant hormone was identified in 1979 and called Brassinosteroids. Brassinosteroids, BRs, are a class of growth-promoting steroidal phytohormones. BRs control almost all aspects of plant growth and development and also play a significant role in plant adaptation to biotic and abiotic stresses. The major role of brassinosteroids in plants is the promotion of stem elongation that is independent of gibberellins. Brassinosteroids also affect crop yields, stimulate uptake, enhance xylem differentiation, enhance resistance to chilling, disease, herbicide, and salt stress, promote germination, and decrease abortion and fruit drop. They also have the potential to be used as an insecticide and in plant tissue culture. You now have a better understanding of plant growth regulators and their many uses in agriculture today. You learned the definitions for a plant hormone, plant growth regulator, auxin, gibberellin, cytokinin, abscisic acid, ethylene, and brassinosteroids. In addition, you know how each of these 6 classes of plant hormones was discovered and the key names of scientists associated with their discovery. Each of the 6 classes of plant hormones is involved in key physiological processes in plants. On the slide, you can see a table summarizing the key physiological functions in plants. Plant growth regulators can be used in combination or separately to manipulate nature for agricultural production.